Thanks for taking the time to chat with me. My name is Elizabeth Finn. I am an assistant professor at the Oklahoma Medical Research Foundation. And what that basically means is that I am running a lab, I am mentoring graduate students, and I am mentoring postdocs, as well as other trainees.
Can you tell us a little bit about what your lab does?
What I'm the most interested in studying is how the genome folds. Because if you stretched your genome from end to end, it would reach about 10 feet. And that needs to fit into every nucleus in every cell in your entire body. And that's a very small volume. And it turns out that the way you do that really matters for cellular function. Because genes, if they're put into different places in the nucleus, will be either transcribed or not transcribed. And when you get misorganization of DNA within the nucleus, you tend to get diseases like cancer or like neurodegenerative diseases. And so I'm really interested in how you get from something so big to something so small in a way that keeps cells healthy. During my postdoctoral work, I observed that in addition to that, every cell folded its genome differently. And so in addition to having to do it right, it's not just that there's one correct fold and one incorrect fold. Instead, there are a million correct folds and potentially a billion incorrect folds. And this is a really fruitful area of research for me and something that really piques my curiosity because the plasticity and the variability that we have even within healthy genome folds means that cells can use how the genome folds to respond to signals healthily and to differentiate properly and to respond to the environment. So I'm interested in studying how the genome folds and how that fold varies, because I think that if we understood that, we could have much greater insight into how cells respond to their environment in healthy ways as well as in disease.
That's very interesting. I had never thought about the differences between genome folding between different individuals. Do you also observe differences between, for example, cell types?
It's actually not even just between cell types. So there are differences between individuals. There are differences between cell types. And what we did during my postdoc was we looked at one cell type in one environment in tissue culture where we had subcloned it out fairly recently, and we still saw differences. We still saw variability. So the plasticity of it seems to be truly stochastic at a certain level, that the way your genome folds is a source of plasticity for a normal cell. And any normal cell, even over the course of a single interphase, a lifetime of a single individual cell, the expectation is that the genes will sort of wiggle around relative to each other.
So within that topic, what questions or what projects are you currently following in the lab?
So I have a few major aims right now. The technologies that I primarily use are microscopy-based. So we are physically we're staining individual genes, several of them in a cell, and measuring relative distances between them, measuring distances to the edge of the nucleus. And those techniques are absolutely great at measuring sort of clustering of silenced genes and heterochromatin. And so one of the main areas that I'm looking to dig into in the next few years is following up on heterochromatin silencing and heterochromatin regulation and heterochromatin organization. So I have one project in the lab that someone's working on right now, just doing sort of a survey of genes that are marked by different kinds of heterochromatin, different histone modifications, and looking at whether they have similar or different organizational patterns. I have a project that I'm really interested in and I hope it goes well, where we're doing a screen to see which genes are driving heterochromatin to cluster within the nucleus. Because we see it happens. Heterochromatin loci that are very far apart in the genome tend to be brought together in space. And we think it has to do with certain proteins, such as HP1. But you can remove HP1 and it doesn't sufficiently, you still get that clustering, at least some of it. So we're looking to see what are the other proteins that are driving that clustering. So that's one aim. And I think that eventually where that's going to be going is something truly more functional, which sort of leads me into the second area. One of the things that I'm most interested in looking at is how these patterns arise in development and differentiation. So stem cells and pluripotent stem cells have very little true heterochromatin, and their genome is much more dispersed. And over the course of early differentiation, as you get your first few cell decisions, your first few germ layers, you see the arising of heterochromatin, and you see these genes being brought together in space. So I want to look at the timings of that. I want to see if the genome organization changes before other lineage-specific markers, see what's causing what in that. The other thing that we've seen is that in cancer, you get less heterochromatin and more variability. So tracking these kinds of regions in cancer cells is another big aim of the lab. And then the last thing is, as a field, the sort of classic example of genome fold leading to function is this idea of a promoter-enhancer loop, where you have a gene that's regulated by its promoter, and somewhere hundreds of thousands of base pairs far away in the genome, there's an enhancer element, and it forms a loop with that promoter, and that drives transcription at that gene. And that model is coming under a lot of fire recently. It's actually a controversy in the field. It's really cool. And one of the reasons I think that we're in this place in the field is that none of the techniques that currently exist have the capacity to truly measure whether the loop is forming and transcription is occurring. There's either a resolution limitation, either genomic resolution or spatial resolution. There's a timing limitation. There are techniques where you can get DNA or RNA, but not both. And a lot of the techniques are sequencing based, and so they don't give you a direct spatial measurement. They rely on an approximation of a biochemical reaction, which is a big limitation. And so I have a few ideas of how to develop some techniques that would allow us to actually measure that. And I'm really excited to start building those tools.
Yeah, that's very exciting, and it seems like it would have a great impact in the field in general.
I hope so. We have to get it working first, and that's going to be a really big lift. but if we do, it would be really, really fantastic.
Very cool. So, going back to the very beginning of this conversation where you said that one of the roles that you have is being a mentor to students and other people. So how do you view this role? And can you talk about your mentorship style?
I think at best, as a mentor, I am a healthy enabler. I think that there's a big difference between mentorship and management. I think my goal as a mentor is to help you identify your goals and to identify the places where our goals overlap and to support you in working in those areas. Obviously, if I just let someone do whatever they want, grad school can end up taking 15 years and not winding up with a PhD because there are so many different paths that you can go on. But I also think that the whole joy of science is in asking your own questions and finding the ways that curiosity motivates you. And so my lab won't be as effective unless it is motivated by the curiosity of the people within it. So that's why I think it's a healthy enabler.
what would you tell someone that is interested in the work that you do or in forming part of your lab? How would you describe, or what would you tell them about your work?
I think that it's a really exciting moment to be studying genome organization because there are so many new tools on the market, and there are so many ways in which the paradigm is shifting. So in the past 8 to 10 years, we've been going through this consortium project called 4D Nucleome, which was attempting to do what the Human Genome Project did for DNA sequence, only for this three-dimensional structure. And as a result of that project, we have many more technologies than we did. We have a lot more people working on it. And we're starting to get those technologies to work together in a way that they really never have before. And that's a huge advantage. And it's a really exciting opportunity for us as researchers to do some really cool work. But it also means that there is no established path. It means that if you do an experiment, you kind of have to invent your own or develop your own analysis tools. You have to be very careful and very rigorous to make sure that your experiments actually are measuring the things that you want them to measure, instead of just trusting blindly into a pipeline or a experimental methodology that's been done for years and years and years. So it's a huge advantage because all of a sudden we can see things that we just never could before. And it's also a source of difficulties because there's no map for where we're going from here. And we really have to make those choices ourselves.
It sounds challenging, but also it sounds like there's a lot of opportunities for growth and a lot of work to be done.
Absolutely. I love it as a field because it's a very quickly growing field, and I have found it to be a very welcoming field as well, where we know that there's far more work to do than any of us could do on our own. So we want more people to be studying these systems and asking these questions.
How did you originally get involved with the field?
When I was an undergraduate researcher, when the dinosaurs still roamed the earth, I worked in a lab as an undergraduate, and we were making transgenic mouse cells with a custom insert that we could stain fluorescently so we could track it in live cells, and where we could also very easily sort of recombine in and out various genome regions. And the idea was that this system would allow us to take an arbitrary gene or an arbitrary sequence, dump it into this tagged site, and see where it went in the nucleus. And this idea that where you are in the nucleus matters, and that instead of being this passive linear structure, DNA was breathing and alive, and not only breathing and alive, but that like breathing and alive in such a way that it fit with all of the things that you learn in biochemistry and in organic chemistry, where shape is function. That inspired me, and I fell in love. And I did have a bit of a circuitous path because at that point I told my direct mentor, the postdoc I was working with, I want to study genome organization for the rest of my life. And she very rightly said, Elizabeth, you're 21 years old. There is no way you know what you want to do for the rest of your life. That's just not possible. So I went to graduate school, and I went to graduate school in a place where there was no one studying this stuff. And I studied DNA methylation and genomic imprinting and weird epigenetics, and I liked it, but I didn't love it the way I loved this genome architecture stuff. And for most of my PhD, I was sort of looking for ways to get back to, but what's the shape? And so I switched fields back during my postdoc, and it was such a good decision. And I get to do this now, actually forever. And yeah.
I appreciate the way that you talk about it. It's inspiring. And I also like how you mentioned that in grad school, during your PhD, you weren't exactly studying what you are currently studying right now, but I'm imagining that you still gathered tools and knowledge that are helpful now. So how does the work that you have done in the past influence the work that you do now?
So in graduate school, I was doing a lot of RNA sequencing, and I was doing a lot of bioinformatics, and it was all in these developmental biology, mouse development, and early mouse development models. Um, and it meant that I had a computational framework and a statistical framework that when I got to the point in my postdoc and we had these absolutely massive image, like pictures of cells, data sets, I was able to apply big data statistics to them in a way that made the data far more legible than it would have been if I had been sort of sitting at my computer and hand measuring things. Um, and so that was a big strength. And I encourage every single graduate student to learn some computational skills. I don't think that you can be a good basic biologist these days unless you can do at least some computational skills. It doesn't have to be your main focus, but you need at least some. The other thing is that because I was doing those developmental models, that interest I kept, and that interest I really liked. So during my postdoc, we were working on cancer cells. We were working on sort of cell line models that didn't have much relevance to development or to disease necessarily. And one of the things that I would love to do in my lab is now bring that back to a model of how an organism develops and how a disease progresses.
This is just for personal curiosity, but do you think that the way that the chromatin organizes is predetermined, or do you think that it is like influenced by different factors throughout someone's life, or is that the question that you're still asking?
Well, I mean, a little all three, and I'm going to throw another possibility at you. So my big thing, like if I have a brand, to the extent that I have a brand, what my brand is that the variability, the plasticity might be the important part. So for this in particular, the predetermined part might not be a genome fold. It might be the wiggliness of the two regions. And so then if that's the case, then the fold is absolutely not predetermined. It's not determined by the environment. It's the wiggle that's responding. And so the fold itself is truly random. It's a roll of the dice. But that wiggle is somewhat genetically determined. It's largely environmentally determined. And so because you have the fold potentially leading to transcription, but the wiggle is what's being controlled, you get this really lovely, like movable dial of regulation where you're regulating that wiggle, and it means that you're regulating the frequency at which an event happens. And so you can start this cascade with like incredible plasticity and incredible robustness by, instead of saying this is the shape that it has to be, instead saying this is how much flexibility we're giving you around the average shape.